Right, this is question 1. A diagram shows a transverse section of the spinal cord. Draw and label the position of the sensory neuron and a motor neuron and label the cell bodies. So you can see there's going to be 3 things that you need to do to get these 3 marks. And which neuron to put where? Well, this ganglion here is where a cell body is going to be, so that's going to be our sensory neuron. So our sensory neuron is going to start off wherever outside, and it's going to go up here, and we're going to have the cell body there, and that's going to go into the grey matter. There might be an intermediate neuron in here, but we don't need that to show you that just at the moment. So that is going to be label the cell bodies. So this is going to be our sensory neuron cell body, and the cell body for the motor neuron is actually going to be in the grey matter there, and then that's going to go out this side, and then that's going to, there might be a muscle or something like that. So this is the ventral side, and this is the dorsal side. And again, let's not, the instruction is cell body, so that is the motor neuron cell body. So, and this is in the ganglion there for the sensory neuron. So 3 marks there. That's AO1, just recall of information. Hopefully you've learnt that reflex art. Part A2, when a person touches a hot object with their finger, they will move away from the heat. State the advantage to humans of having this type of response to an external stimulus. This is why do we have reflexes, really. Again, AO1 should just know why you've got reflexes, and it's to protect from injury or harm. You don't want to be keeping in contact with a hot object. So to prevent injury, harm, or further injury. Part B, Snyderia, and you should immediately recognise this diagram of Hydra. It doesn't say it's Hydra. Cell bodies consist of a jelly-like substance sandwiched between 2 epithelial layers. Epithelial means on the outside of something, and it shows the nervous system. State the name given to this type of nervous system. Again, just AO1, straightforward. Have you learnt it? Nerve, net, and you need both those words there. Part 2, explain why the non-myelinated neurons of Snyderians result in slower impulses in mammals. And the clue is here, they're non-myelinated. So we must have to compare these to our knowledge of what myelination does. So this is AO2. We're taking our knowledge and we're applying it to this situation. So in this case, these are non-myelinated. Our impulse must travel along the axon with all of those things that you know about the gates opening and closing, the pumps working, and so on. That would be in our Hydra. In mammals, so let's draw a little stick man, there's a mammal. We've got the axon, but we've got myelination, the Schwann cells that contain myelin, kind of fatty substance, and the impulse can jump from node to node, from each node of Ranvier. So you only get depolarization at the nodes, and that's quicker. So that's what we need to explain. So let's talk about the Snyderian. So I'm going to write the word there just so we know which way around we're talking. The impulse must pass along the whole length of the axon. The impulse, or the depolarization, or whichever, how you're going to say it, must travel along the whole length of the axon, or the membrane. And then we're going to say, why is this slower than in mammals? Because in mammals they have a myelin sheath with nodes of Ranvier, and this causes saltatory conduction. Saltatory meaning kind of dancing, really. The saltatory conduction, or you describing it going from node to node, enables saltatory conduction, I'll put in brackets, node to node, which is faster. You could have put the reverse argument about Snyderians not having that same thing. So that's 7 marks.